Hi, everyone. It's 11 o'clock here, so we're going to get started. Thanks, everyone, for joining us today for our webinar. My name's Jackie Carville, and I'll be coordinating the webinar today. I'm here with Matthew Kaiser, who will be going over some microRNA workflows within DNASTAR's Lasergene Genomics suite. You may have noticed that your phone has been muted. However, we do encourage you to ask questions along the way. To ask a question, just type it into the chat dialog and select Send to Host. I will then direct these questions to Matt at breaks to be answered for the entire group. If you need any assistance or have any questions during this webinar, you can send a chat message to me, email me at webinars@dnastar.com, or tweet us at the Twitter handle @DNAStarInc. And with that, I'm going to turn it over to Matt.
Thank you, Jackie, and thank you for joining me this morning. So today we're going to be talking about some miRNA tools in the DNASTAR Genomics suite. And I've had a number of customers in the past year or two that have miRNA data and really have wide-ranging needs, and depending on the workflow and the organism that they're working with. And so we thought it would be a great idea to do a webinar that shows you some of the flexibility and the different options in the DNASTAR software packages to work with this type of data. And so this will be a little bit different than some of the webinars in the past that, there's generally one workflow that we start, from point A and go to point B. Here we're going to have some optional workflows. So I would imagine there will be some questions. So I'll try to explain the different workflows in as much detail as possible. But certainly, if you do have questions, send them in to Jackie and I, and I'll try to clarify and answer your questions directly. So a little background on the company. DNASTAR is located in Madison, Wisconsin. Like most of the country, our weather has not been sunny and warm. We're in cold rain, but at least the ice is off the lake as of a couple weeks ago. So we're moving towards this kind of view soon, I hope. So DNASTAR was founded by Dr. Fred Blattner at the University of Wisconsin-Madison in the genetics department. And Fred and his lab are pioneers in genomics. And back in the 1990s had sequenced the E. coli genome. A number of the scientists that are on this science paper still work for DNASTAR and provide valuable input and direction on our product development. And really our goal since the beginning has been to develop powerful tools to put in the hands of researchers so that they can work with their sequence data and do their own analysis, using their personal computer, and of course getting support from a company like DNASTAR. So over the past 30 years, DNASTAR software has been cited more than any of our competitors. And so I like to show this, just a bar graph to show that, over the past 30 years, we've had almost 10,000 citations in peer-reviewed publications. And so this really is research-grade software. We've been in this business for the long haul, and it's a difficult business to stay in business for a long time, and that really requires great software and really great support that goes along with it. So speaking of our support, there's a number of different ways that we support our customers. We have, for instance, on our website, over 100 videos that vary from 30 seconds to just a few minutes that cover a variety of different topics. We haven't had one for microRNA, and so we're doing this webinar today. So we'll have a webinar on microRNA analysis and then possibly some smaller, shorter, more focused videos on this workflow as well. So under our support section, you'll see, again, all these short videos that help our customers tremendously. We also have webinars like we are doing right now. Myself and Jackie and some of the other support people here will do webinars on a variety of topics. And it looks like we've reorganized them recently by year, so we go back to 2012. And you can see that the most recent webinars, I did one on advanced sequence editing and annotation about a month ago, and we've done a protein webinar and some variant detection for rare diseases. And so, again, if you have other workflows that you're interested in, under the support tab, the webinar schedule is a great place to get about a 45-minute in-depth discussion on that workflow. So today we'll be talking primarily about the Lasergene Genomics suite. The genomics suite is going to be comprised of 3 programs that we're going to be looking at. That will be SeqMan Engine, ArrayStar, and then also the SeqMan Pro module for visualizing the assembled data. Of course, DNASTAR offers other software tools that focus on things like sequence alignment. So in the evolution suite, we can do sequence alignment and editing in phylogenetic trees, and we have some new software there in Megaline Pro that will be released this spring that expands that functionality, as well as some new tools for protein analysis and protein folding. So again, the entire DNASTAR package offers very wide-ranging functionality. Today we'll be talking about assembling next-gen sequence data and doing downstream analysis. So one of the questions that will often come up is what type of hardware do you need along with the software to handle this type of data? And our software has been designed to take advantage of increasing power of desktop computers. So as these computers have become more powerful, we've been able to do much larger data sets on modest computers. And so for the data sets that we're looking at today, a laptop is sufficient, for the microRNA assemblies, and really a computer that has a solid processor, a 4-core, 8-core computer, 16 gigs of RAM, maybe in some cases up to 32 if you have large, deep assemblies, and really what's important for the alignments to reference genomes is having scratch disk space. And that the amount of space needed varies from about 1 to 4 terabytes of space if you're going to be aligning to eukaryotic genomes, for instance. So today we'll be looking at an Arabidopsis genome. 1 terabyte is more than enough. You could probably go with a quarter of that space to handle Arabidopsis. But we need the disk space to give us the really nice, fast performance. And then, of course, storage for input and output files. So ideally, it's a 3 hard drive setup. 1 hard drive to run the operating system and the programs, 1 hard drive dedicated for a scratch disk, and then, of course, a hard drive for storage of files. So that gives you the best possible performance. So the different workflows that the genomics suite can accommodate are wide-ranging. So again, DNASTAR software has been designed to be as flexible as possible and accommodating to all different types of workflows. And that's really highlighted in microRNA analysis because, again, depending on what your needs are, you may need different components of almost all of these workflows to do a full analysis of your microRNA. And so the workflows, can be as diverse as de novo transcriptome assembly to targeted resequencing and exome resequencing to RNA-Seq to viral host integration. So the algorithms are flexible and can accommodate different types of sequence input data. And of course, the algorithm can flex to accommodate de novo or templated assembly. So today, of course, we're going to focus on microRNA. And there's multiple sub-workflows with microRNA, and this is where it can be, because some of you have different needs. Some of these different sub-workflows will pertain to you more than others. So I'm going to try to just discuss the different options that you have in the software, and there are probably some other sub-workflows here that I haven't imagined or worked with customers on. And that's where you may have some questions. If you have a particular data set, we may have some other modifications to these workflows. So there's 3 kind of workflows that I'll be looking at today. The first of these is alignment of microRNA of data to reference sets to quantitate and compare different levels of microRNA sequence data. So, for example, you may have multiple individuals or multiple tissue types or developmental time courses, and you want to assess whether or not the microRNA levels are in flux or changing between the different experimental conditions. And so we'll do that analysis on ArrayStar. ArrayStar has our quantitation tools. It can take an alignment, has a aligner built in that's very similar to SeqMan Engine. It can align sequence data, map it, and use that mapped count of reads to do a quantitation. So we'll start there. And then we have doing de novo discovery. So you may have an aspect of I want to know all the known microRNAs, but there's also always new ones being discovered. How do I collect the sequence data that would represent potentially new microRNA species? And even if you're working in the main model systems like human and mouse, there still are new microRNAs being discovered all the time. In some of the non-model organisms, or today we're going to look at Arabidopsis in plants, where there's many new microRNAs that are being discovered all the time. So we can work with the data that does not align to the miRNA reference sets. We can collect those reads. We might de novo assemble them. So in cases where you don't have a reference genome to work with, I can de novo cluster them together and find new microRNA sequences. In other cases, like Arabidopsis, I may have the reference genome to align back to, so I can map these potentially novel species back to the reference genome using SeqMan Engine and get an alignment in a BAM file I can go and look at and interrogate and visualize. So this is where we would use the SeqMan Engine assembler. And then there's some other ways to work with microRNA data. So for example, if I have RNA-Seq data and miRNA data for the same RNA data set, I can compare changes in gene expression to miRNA that's detected, for those genes, for instance. And that will be done again in RNA-STAR, where I can compare RNA-Seq expression values with peaks, areas where miRNAs are essentially aligned near genes of interest. So RNA-STAR has the unique capability then to combine different types of sequence data together in one analysis. So we'll spend some time looking at that as well. So at this point, I'd just like to jump out of the PowerPoint and start showing some of the software. And so the starting point today is going to be RNA-STAR. Now, usually our workflow will start with SeqMan Engine. We'll run an assembly, look at it, go to RNA-STAR. But with microRNA, it's a little bit different. We usually are going to start with our RNA-STAR. And I'll launch RNA-STAR here. And so RNA-STAR is a program that has really evolved over the last few years. It was originally designed for microarray analysis, so it could import sequence data, or chip data, and from in essentially large tables of data, and there would be a gene list, and each gene would be assigned an expression value. And so RNA-STAR could bring in multiple different sets of data, compare them statistically, import annotation information to associate the annotations with the genes, and you could get a nice gene expression plus gene function type analysis. Soon after we released RNA-STAR, next-gen sequence data started appearing, and so we designed QSeq, which is a mapping algorithm. QSeq is actually a pre-runner to SeqMan Engine. It's a very similar algorithm for mapping alignment sequence reads to a reference genome. The difference with QSeq and SeqMan Engine is that QSeq does not produce a gapped alignment. It is not producing a BAM file that we can go and visualize. It is taking reads, mapping them to a reference, and building a count. So it's an extremely fast, robust mapping algorithm. It can go through massive amounts of data very quickly and then give us a count, which can be normalized, so we can then use all the tools that we'd use for microarray to do things like gene expression analysis. So that's kind of the background here on QSeq. Now, when we start looking at other workflows, so RNA-Seq, we map the reads, we normalize the counts, and we do expression analysis. There's also some SNP analysis that is, comparing, a huge, we could have 100 exomes where each one has thousands of SNPs, and compare those data sets and find SNPs in common or unique to the different groups, and copy number variation. But if we go all the way down to, there's a microRNA workflow in QSeq as well. And this workflow is, what it's doing is it's aligning microRNA data typically to a reference genome. And what it's doing is where those reads align, there's a peak of alignment, and where those reads pile up. And when we align to that reference genome, we can RNA-STAR can detect the peaks and tell you what gene the peak occurs in, or it can tell you the gene that's upstream or downstream. So it's similar to a ChIP-seq kind of an algorithm where with ChIP-seq, you're looking for reads that align in peaks, and you want to know, is that related to the potentially the gene that's in the neighborhood for regulating that gene expression. And so the same is done with microRNA if you want to look at potential regulation of genes due to the presence of these microRNAs. So that's one way that we can use, and we'll look at that as our third workflow, as how we can look, compare the the microRNA peaks with changes in gene expression. If we want to look directly at microRNA levels and changes in levels between different experimental setups, we can treat this like an RNA-Seq project. And I'm just going to click on RNA-Seq here, and we get an interface to set up the project. And so I can add files, and these files now are going to be, again, I have a Arabidopsis microRNA data set, and there's 4 different conditions, and there's FASTQ files here. And so I can add FASTQ files then for each experiment. I'll just add one more here. So here's 4 different experiments, and again, this is Illumina FASTQ data, and I'll just click Next. And it's going to ask me how I want this processed. And because in this first experiment, I really want to know, are there different levels of different species of microRNAs between my 4 experiments? And so we're going to use the QSeq processing, and this explains in some detail what it's doing. It's going to be a normalized signal. We're going to align reads and then normalize for differences in these FASTQ files have different numbers of reads in the file, so we have to normalize for that. And so we're going to use an RPKM normalization. And then this big box here is asking me, what do I want to quantify against? And so with microRNA, there are several different options. It could be a genome, but typically we're going to go to a website like miRBase and download a set of microRNA sequences. So we can go to miRBase, and there's lots of information here, but I'll click the Browse button. And I can browse miRBase by species. And you can see here we're working with Arabidopsis, so I'm going to click this link. And I've got, for the brassicas, I've got 5 different options. And so I'm just going to click on, and we'll let this. So this is a great resource now to find the miRNA species for your particular organism, and it's a little bit blurry here, but and so I can go to this. There's 298 of these Arabidopsis microRNA reference sequences, and I can go here and select these sequences and fetch all of them. And so I'm going to use these microRNA reference sequences now to quantitate against. And so I've already downloaded this file. I also converted, let's take a peek in here. Let that come up here for a second. Sometimes when we download microRNA reference files, we have uracils instead of thymidines, and so I actually converted them from U to T to make the analysis more streamlined. So I'm going to load in these sequences. You can see there's it's a FASTA file. There's 298 sequences, and they're not annotated in any way. I downloaded the FASTA file. So ArrayStar is just going to align to the whole sequence. It's not going to look for annotations that are subgrouped or within these small sequences. And so here's my 298. There are some advanced options here. There is something very important in these advanced options, and it is the unassigned reads. And so the expectation here with my data set is that with many plants, and especially if it's a species that's maybe not identical or a close species, I'm expecting that when I align my reads to this miRBase data set, that only 1% will actually align, and I'll have a large pool of reads that it can be other RNA that was isolated in the prep that's just noise, that's just junk that I might not be interested in, but there could also could be a whole lot of other mRNA species in my sample. So I want to collect that data. I don't want to throw it out. So I'm going to save this, up to my desktop in my folder. So you have to go to the advanced options and tell RNA-STAR that you want to save the data that does not align to the reference sequences. So these will be the unassigned reads then. So that is very important to check that option. And then when we click next, it's going to be ready to go. And it's fast, so you can see it's again, this algorithm, like SeqMan Engine, uses disk space. So it's going to use hard disk to process temporary files. It can go through millions of sequences, just in a few minutes. So you can see it's going through. We're not going to wait for this to finish though, because I've already done this. So like a cooking show, we will go right to and I think I can just close here to what I had assembled. And so here's my 4 experiments, and I name them. I just right-clicked and name them, change the name from the FASTQ file to the experimental name. And so I can start to look at this experiment. So I can highlight an experiment and get some of the attributes. So if I click on the first experiment, you can see there's a list of experimental attributes. And I can see the notes. I can see that we normalize the signal with RPKM. 158 genes are missing. What that means is that sequence data align to only about half of the miRNA reference sequences, any sequence data at all aligned. And then I get some statistics. So I can see that, in this first experiment, we had how many reads is that? About 10 million reads, and only 2,437 were uniquely mapped. So again, in this kind of a prep, there's a lot of reads in there that aren't specific to miRNAs. So we're kind of fishing through the data to find those reads that map to our reference set of miRNAs. And so I can see a fairly low number mapped. So there's a big potential for novel miRNAs that did not map to this set that we used. Okay, so I have my 4 experiments. I can now do things like go to a gene table, and the gene table now has the list. So this is the name of the miRNA reference sequence, and I can see my 4 experiments, and this is an expression value that's been normalized, so it's in a log 2 value. And I can hover over any cell here to get the raw information about the different data. I can also remove these columns and go to raw counts as well. And so I can look at the different raw counts, sort the columns. So I can see that for some of these experiments, we've got 352 reads aligning to some of the more abundant species that are here. I can also do things like go to a scatter plot and compare. So I can compare 2 different conditions to each other and then start applying filters. And again, we ran this through an RNA-Seq, so we've quantitated the aligned reads to the reference, so we get an expression value then that is related to the number of reads that have aligned. So there's different ways I can go through and filter and say, well, show me all the genes that are at a 2-fold change. And so I can click that link and it highlights them, and that's when I go back to my gene table. They're checked now. These show a 2-fold change. Or I could run something like a heat map. So there's a number of different ways I can run a scatter plot. And I have some quick links in the bottom right here where I can do a clustering of the genes, generate things like heat maps between my conditions. So again, if your experiment is that you've got different experimental conditions and you want to compare relative expression or abundance of miRNA species, you can do that directly here in RNA-STAR. Okay, so we could see again from the experiment list that most of the reads, the vast majority did not align to our reference set. So we're definitely going to want to interrogate, why didn't all those reads align, and is there more data in our data sets that we can extract. And so one way to do that is to focus on the reads, the unmapped reads. And we can do that a couple different ways. One of them is just to do a de novo assembly. And we can take that output data, and I'll show you what that looks like. So these are these unassigned reads from sample 1, 2, and 3. I did not, let's see, oh, and this would probably be 4 here. And so I have 4 FASTQ files that were saved out, and you can see they're pretty big. They're almost a gig in size. So they're sizable data. And if I go back to the original data files, they're about 1.5. So we've got the bulk of the data not aligning to our reference set. So I can go back and take this data and do 2 different things with it. I can do a de novo assembly, or I could align this data back to the Arabidopsis genome and look for areas that have aligned and kind of interrogate and see, well, is this all miRNA data, or is there other kind of data or noise present in the sample? So I'm going to launch SeqMan Engine now, and SeqMan Engine gives me a wizard for setting up different projects. And if you've seen other webinars that we've done on next-gen topics, that SeqMan Engine accommodates all different types of workflows. And again, the difference between SeqMan Engine and ArrayStar is that SeqMan Engine produces a gapped assembly. The reads are maintained, saved to a BAM file. We can view them in SeqMan Pro. We can do SNP analysis, and visualize. So when we're doing de novo assemblies, we really want to look at the aligned data. So I'm going to create a new assembly project, and miRNA. Pick that workflow, and I have 2 options, de novo or alignment to a template. And so I name my project and save it out here. And now there are other workflows. I won't explain all the de novo pieces in detail. There are other webinars and videos that talk just about de novo assembly. So forgive me if I go through here too fast, but we're going to pick the editable SeqMan Pro format. If your data set is a lot larger, which it very well could be, so imagine that you've got 500 million Illumina reads, and you're going to be de novo assembling a big chunk of them. I might switch to a BAM format, which there's no limit to how big the project can be. You can have millions or billions of reads in the project. However, in a de novo assembly, we always have to keep in mind that de novo assemblies are RAM limited. And so it depends how much RAM you have on your computer as to how much data you can assemble at one time. And I find that with microRNA, 10 million is even more than what you typically want to assemble at one time. So I'll just show you, I'll set up, these are short Illumina reads, and I would do 1 sample at a time, like this. And the reason for that is I don't want to throw all the data into a de novo microRNA assembly. And the reason for that is there may be some species or some elements that aren't even microRNA, but maybe ribosomal RNA, for instance, or that can be extremely abundant in the sample. And so I don't want to throw all the data in and assemble it and have a contig that is literally over 1 million sequences deep, which happens quite frequently with microRNA. De novo assembling contigs that are 1 million sequences deep is really inefficient, and so it'll make RAM spike and maybe will cause a problem with your de novo assembly. So when I do microRNA, I'll set up 1 sample and really scale back the assembly and limit the number of input reads. So I might say, I want to take a look at this quickly. I'm going to do a pilot run of just 10,000 reads or maybe just 100,000 reads. And I want this to finish up within an hour and then go and look at the assembly and interrogate it and see, well, what's in this? Is it just a bunch of scraps of RNA that were in my prep? Would I have a not very specific prep, or do I have all microRNAs here? So I would scale this back and just stick with, I wouldn't worry about scanning too much, but we might come back to this. I'll do this pilot run of 100,000 reads, and if I see that there's a lot of ribosomal RNA, maybe I set up those ribosomal RNA sequences of contaminants and pull them out And so you might clean up the data by doing a pilot and use these contaminant scans to pull out some of the noise to help the de novo assembly. And again, there's all different other scans we can do. We do quality trimming by default. Again, de novo assemblies can take more troubleshooting to kind of fine-tune them to get the results. I won't go into all the details here, and so we're going to do a let's see, a minimum match percentage. So of all the parameters that we have here for microRNA, I oftentimes will increase the minimum match, and that is the percent that the read must match the consensus within that contig in order to align and cluster. And because microRNAs can be quite similar, they can be quite similar to each other. I might make this more stringent and say 97%. You know, I want a stringent, clean contig with little mismatch. I'm going to bump that up, and then I might also remove, depending on my data set, if I've got 500 million reads or a huge number of reads, I might say only show me those species where there's at least 10 sequences aligned. If I drop this all to 0, then you might get tens of thousands of little 2-sequence contigs. So a little filter here is nice, and then we're ready to go. So de novo assembly, we click assemble, we watch the task manager, and I'm not used to Windows 8 yet, so let's see, more details. So this looks, so you can keep an eye on SeqMan engine, and when it's doing a de novo assembly, it's going to be primarily RAM usage. So and you can see this little data set finished really quickly, so because I only loaded a few sequences. And you can see when it's, what we have here is the log file, so I can see what version I use, the parameters. This is the 100,000 sequences. If there's an error here, you can export the log, send that to DNASTAR. We can troubleshoot, and you can see it's done, and I can click Next, and it gives me the project report, which tells me the metrics of the assembly. So this is common with all de novo assemblies, and I can launch this in SeqMan. And so now we're going to the third program here, and SeqMan is for next-gen workflows, is the visualizer of the assembled next-gen data. And it can work in two formats, the editable .sqd file, which is limited to about 10 million reads, or the uneditable bam.assembly file that is unlimited in size and scope. And so this little de novo assembly, I can sort the contigs, and I can see, oh, look, there's one here that's got 2,500 sequences in contig 3. And I can double-click on the contig and go and take a look at it. And as you would expect, it looks like a little short, potentially an miRNA species. Maybe it's a novel one. I don't know for sure, but I can blast them, highlight a few of them, and blast. And wait for those blast hits to come back. And you can see the little globe that's spinning here. That means it's retrieving results. So we'll come back and look at this, because sometimes with webinar running too, the blast spools back a little bit more slowly. So we'll come back to that. But so this is a way to de novo assemble. I should show you another thing. So say we find and we determine that by looking at this assembled data, and we blast them, that, oh, these are new microRNAs that were not in my database. So these are new to my organism. I can select multiple contigs, export the consensus sequence. So I can save the consensus out into a single file. I can also name the contig something else. So if I right-click here, I can call this, miRNA, and give it some kind of a number or something. All right, so I can identify them, name them, and then export them out by selecting contig, save consensus, single file. And the consensus then can be include gaps or not. So depending on what format you want, typically we want an ungapped consensus, and I can save this out as a FASTA or GBK or .seq file. So that's a kind of a typical workflow for discovery mode. Now it looks like our blast is coming back here. So let's see what we get. And so that's really tiny. Let's make that bigger here. I always forget font size. That should be better. So the blast hits from SeqMan, there's a couple things with little short sequences like this. Usually the identity, you can see it's saying 100%. And we also get things like query coverage and E values, but it's really this query coverage. You want to kind of expand this column a little bit, and it'll say what contig has been blasted. And of course, there's multiple blast hits now for many of these contigs because they're well represented in the database. And so I can, let me make this a little bit bigger here. Let's see here. Can't quite get to my window here. There we go. Okay, so I can scroll through and see, there's contig 3, contig 1. It's still spooling results, so it's acting a little bit sluggish. So I can see the description then of what what database sequence we're hitting. And it looks like we've got first contig 3 looks like junk. It looks like back clones, and the P fossil vector I think is a cloning vector using some Illumina prep. So it looks like the contig 3 was just noise, and I'm going to sort it. It looks like our search is done now. At least part of it was hitting. Here's contig 1. We're hitting some Arabidopsis, and there's some microRNAs that we're hitting. So contig 1 is hitting some microRNAs. And so it's a nice tool to kind of sort through and figure out, which of these are likely microRNAs, which ones are some kind of noise. Now, the other way to work with these novel sequences is if you have a reference genome, I can take the same data and just align it back to the reference genome. And so that is, again, a SeqMan engine function. So let me just launch SeqMan engine again. Except now we're going to do microRNA templated assembly. And I'll call it novel miRNA, let's see, genomic, something like that. We'll pick our output folder. And now you can see the interface, because this is not a de novo assembly, but templated. The hardware requirement changes a little bit, so it's asking us, where do we want to process our temporary files? And this goes back to we need scratch disk space to process both the query data and the template data. And there's links to our website, and again, there's that explain, like my slide did, what kind of hardware you would need. Now, Arabidopsis is a small genome. The scratch disk space is minimal that we'd need for aligning to Arabidopsis. But if you're aligning to a human genome, then you want to make sure you've got your terabyte of space for these files. And so we'll click next here. Now we input our template files. I'm going to go to our website and download a genome template package. So this is a great place for model organisms to download this package that contains both the reference chromosomes and the dbSNP databases for that organism. So you can download the package. I've got an Arabidopsis here already. Okay. And these are, again, short Illumina reads, and I'm going to go and grab the unpaired reads. So these are all, again, the reads that did not map to the Arabidopsis microRNA reference sequences. So I'm going to map them to the whole chromosome sequences now and take a look at that. So you can see it's a simpler interface, but I might also want to be stringent here and say 97%, and it's ready to assemble. So really limited parameters when it's a mapping or alignment to a reference genome. And then we're ready to align. And again, when this assembly would take maybe 10, 15 minutes, when it's done, we can open it directly in SeqMan Pro. I've already got that project here. And so one of these, let's see if it's this one. Yep. So here's the novel microRNA assembly. And we can see these are the chromosome sequences for Arabidopsis. And I can sort by length and double click. And here's our view. Now we have a reference genome, and you can see some of the aligned reads that are here. And so there's different ways to navigate this. I might go to a strategy view, and a strategy view is like a genome browser type view, and I can zoom in. So there's a gene, and I can see. I'm going to expand here. There's a peak of coverage there. And what is that? It just looks like a repetitive area. So there's different ways to navigate. Usually with microRNA, because the data, these are sparsely located across the genome. These are the peaks. So these might align within genes. It may be upstream or downstream from genes. Many genes aren't going to have any microRNA regulation that's detected. And so another way to navigate is to do a coverage report. And the coverage report is sort of like a peak finder. It's going to say, well, here are areas where we've got peaks of coverage, and so I might want to just, create this report and navigate through my assembly to find areas where there's significant depth of coverage. So this is a way that I can go and look at. So there's a depth column here. I'll click it again. And so I'm just expanding the columns here. So what we're looking at now are different regions of coverage, and you can see the depth here is in the thousands. And they're small little areas, 32, 22. It tells me the location, and also if it's within a feature. And so I can double-click on this report and go right to that spot in the assembly. So it's all interactive, which is great. And so I can go and look at the alignment view, and oh, look, there's an annotation there, and there is a non-coding RNA feature, a microRNA. So interesting, that particular microRNA wasn't in my database that I use. So I would maybe want to go and look why it wasn't near 161 in my database that I downloaded. And I can see here's the aligned reads, and they're really deep. It's a vertical column of reads there that are thousands of X deep. And so I might work through this report then and look for, are any of these aligning? There's probably some noise, so I can do it pretty quickly. There's another one aligning to a non-coding, so maybe my database sequence is updated, or maybe that's the best I can get and I need to append to it. So again, depends on what your needs are. So I can go through and, here's areas that are aligning, and there's no annotation here now. So maybe this is a novel species. I'm getting this peak of alignment. I might want to go and blast this and see, does it hit any other, maybe from other species or other plants, this is a known or it's close to a known microRNA, and I could maybe find a novel one this way. So again, great tools. In SeqMan, when we align the SeqMan engine, we get this output file, and which is called a dot assembly. So this is a BAM format. It allows us to navigate these big assemblies through chromosomes and look at assembled data. So that's when you'd want to use SeqMan engine. So that's a couple of the workflows. There's one more workflow I'd like to just show you, and that's back in ArrayStar. And that's, if you have microRNA and RNA-Seq data for a data set, you might want to correlate those microRNA values with RNA-Seq values. And so ArrayStar has the capability to make those kind of comparisons. And so, for example, if I bring in my microRNA project and use the microRNA workflow, and what that does is it creates a table that is saying, here are all the alignment points where there's peaks of data. What we just looked at in SeqMan engine, little peaks throughout the genome, and it'll give me a table. So this is a file, import experiment, microRNA. So here's the project setup, and I added my files, assembled, and I created my microRNA experiment. And when I do RNA, I get a peak table. And so this was not for quantification, but just to find where these peaks are. And so this is just a list of 2,865 peaks. And the peak table then tells me a few interesting things. It tells me which chromosome the peak was in. It tells me how big the peak was, which genes are close by, so intersecting or upstream or downstream. So I can find, and I can populate this with, here's the downstream genes, here are the upstream genes. And so I can create a gene set then that says, well, it looks like there's significant microRNA data binding or aligning. So I'm sorting my table so I can see that this raw count, there's lots of reads aligning near a set of genes. And so these might be interesting to me. So I might say, well, I'm interested whenever there's microRNA data aligning significantly. So I might just scroll through the table and just put a cutoff and say, well, if there's 25 or more, I want to know about that. So these genes are all potentially interesting. I can right-click here and say, do a couple of different things. I can show the genes near these peaks. I can copy the rows, but I'm just going to show the genes. So here is the list of genes that are near where I had significant alignment of microRNA data. So I can get a gene set like this. And RNA-STAR also allows me to bring in other information. I can say, well, what about these genes? I can download some annotations. And so we have links in RNA-STAR to databases that contain annotation information that link up with these specific genes. So I can go and download Arabidopsis and import that into my project, which I've already done here. And so I can get some information about the genes. So this is just a gene set now of genes near microRNA aligned data. And I can save this now. So if I want to compare this to RNA-Seq, I can say, well, this is interesting. Now let's go and save this. Remember this set of genes. So this is an interesting miRNA genes. And I could make other comments like greater than 25 reads mapped. And so and you can see in this project, I've done this before in this project. So I can start doing different kind of filters or create different sets from my gene table. And so in this project now I have 3 interesting microRNA gene sets based on where those peaks are. Now if I want to bring in RNA-Seq data, I can do a file, import experiments, RNA-Seq. Same interface is going to come up. I'm going to add my FASTQ files for my RNA-Seq data and my genome and say go, and I will get an RNA-Seq data set. So if I go to RNA-Seq now, I can look at things like a scatter plot, for instance. So in this example, I had 2 RNA-Seq sets, 1 and 2, and these are, again, gene-level expression. We have a whole webinar just on how to do RNA-Seq, so I apologize for kind of skipping over a lot of information about RNA-Seq. But the general approach with RNA-STAR is bring in the data, then apply the appropriate filtering to find gene sets that are interesting, based on their expression pattern. So it might be something as simple as a fold change. Create a gene set where the fold change is 4 or more. And I can make more complicated filters because I might say, well, I really don't want genes down here where there's only a couple of aligned reads. So I can make a more complicated filter or more sophisticated filter in RNA-STAR as well by clicking a link to advanced filtering. And again, so right now I have a fold change, but I might say my expression level should really only be, greater than log 2 or something. All right, and I can do a couple of those kind of filters. I can do. So we have all the flexibility here to do pretty advanced filtering to get that gene set, and then I can just do a search. Say, oh, there's 118 genes from my RNA-Seq that show a 4-fold change and also have a baseline expression of log 2, which can be correlated to the raw count, which might be 25 to 50 reads or something like that. So each time I filter, depending, it doesn't matter how you filter, but you can save that result. So I have 118 genes. I click this box and I say, RNA-Seq 3, and then now because I use the filters in RNA-STAR, the notes here save what I did, how I created that gene set. So I can create as many of these sets as I want, but now what I've done is I have microRNA peaks and gene sets based on those peaks, and I also have gene sets based on expression levels from RNA-Seq. So now if I want to compare them and say, well, are any of these genes in common? Do I have peaks of microRNA potentially affecting the expression of those genes with actual observed expression changes in those genes? And so I can start to make those so depending on how I make the sets, I can start to make those comparisons. And that's where we can do things like Venn diagrams. And let's see if we can get sometimes my webinar fights with this window, but it looks like we're okay. So I can create Venn diagrams. Now the Venn diagram, I can control what I'm comparing, but in this particular example, the A region is the set of microRNA genes that were interesting, and B is the RNA-Seq genes that showed an up or down regulation. And I can look at the intersection. Well, here are 94 genes that had a peak of miRNA data aligned either in the gene or upstream, and also showed a significant change in expression. So then I can, again, in RNA-STAR, I can say, well, that's interesting. Let's go look at that gene set. So I can select that intersection, show the table of those genes, and again, I can bring in the go terms, and maybe I want to do things like look at a gene ontology then, and say, well, is there anything in the gene ontology here that, and it looks like the one that kept popping up with this particular data set was enzyme regulation activity seemed to be significant with these particular sets. So again, and again, these are explained in other areas in our webinars and on how to do more gene ontology. So it's a great tool. So if you look back now, as we wrap up here, depending on what your needs are with microRNA, the Genomics Suite offers all different options for you. Ranging anywhere from quantification against a reference set of microRNA sequences to novel discovery through de novo or alignment back to a genome. And you can also use the tools to compare microRNA to RNA-Seq or other types of data like ChIP-seq and make comparisons between two different data types. So with that, I think I'll wrap things up. I'll be around for the next few minutes to answer any questions that have come up. I don't know if Jackie has any questions for me, but...
Hi, yeah, thanks, Matt. As Matt said, feel free to chat in your questions at this time as we finish up here. We do have one question. Someone wants to know if you can speak as to some of the options available for doing some of these workflows when using non-model plants here.
Yeah. So if you have non-model plants, one starting point might just be a de novo assembly. So it's similar to a non-model transcriptome assembly. What I would probably do is do an alignment to whatever plant is most closely related. So if it's a Arabidopsis, or I would go to let's see, I'll show you back here. I would probably go back and to miRBase and find the most closely related organism and download that reference set and do an alignment and just see how many of those microRNAs align to the most closely related plant. If very few of them align and it's really a novel organism, then you can take those reads and de novo assemble them in SeqMan Engine and then export the consensus sequence out. So I showed that briefly here. But I can go and export the consensus out and start to build my own reference set for that organism. So once you've identified enough novel microRNAs, you can use those as a reference to do things like quantitation in RNA-STAR. So it's starting from scratch, and building that reference set really becomes the number one priority.
Wonderful. Thanks a lot, Matt. So it looks like we're about out of time here today. You can still chat in your questions in these last couple minutes. If we're not able to get to it, we can definitely follow up with you after the webinar with a more detailed description. Additionally, if you think of other questions later, you can also email those to me at webinars@dnastar.com, tweet us at DNASTAR Inc., or contact us via any of our other social media channels. Also, feel free to email me with ideas for future webinar topics. Thanks again for joining us today. We hope you found this webinar helpful. As Matt mentioned, we do have a large collection of videos on our website, so I would encourage you to check those out when you have the time. We're currently scheduling some more upcoming webinars, so I would check back on our webinars page or across our social media platforms for some updates on those, which should be coming soon. Again, you can register for any of these future webinars as well as watch recordings of all our past webinars on that webinars page. Thanks again for joining us, and have a great day.